forest in the Netherlands, the most popular outdoor attraction in the country with more than 200 million visitors a year. Until the 1970s, a large part of the forested area was managed for wood production, which resulted in monocultures. In the last decades, focus shifted towards a more integrated forest management in which ecological, production and recreational values are simultaneously achieved. This shift, together with climatic changes, may have caused the increase of one of the most dangerous animals of Europe, an animal almost invisible to the human eye, the tick. Public health organizations all over Europe are sounding the alarm. Infectious diseases due to tick bites have increased dramatically over the last years. One of the most threatening tick-borne diseases is Lyme disease, caused by the Borrelia bacterium. Annually, 25,000 to 30,000 people in the Netherlands get infected with Lyme disease. Due to the scale and importance of this problem, it is highly relevant to know what factors determine tick population dynamics. Ticks go through different stages during their lifetime: the larval, nymph and adult stage. A question that we can ask, in order to get more insight in tick population dynamics, is how is life stage related to host preference?
Different life stages need different hosts. So larvae are generally to be found on small rodents, mainly mice. The adults you mostly find on large mammals, but most important is that the larvae you find on mice and the adult ticks on the larger mammals.
From this explanation, it is clear that small mammals are of major importance for tick survival and reproduction. The common small mammal species in the Netherlands is the wood mouse. If tick density can be explained by wood mouse abundance, then what is a suitable habitat for a wood mouse?
It's anything with trees with large seeds like beech, like oak, hazelnut, things like that. Also smaller seeds they eat. Forest with insects they eat. They eat tree seeds when they're available and they hide them for later use. But in the meantime, they also eat insects. They eat mushrooms, all kinds of. So any habitat that contains these food sources, in principle, is suitable for wood mouse. This is not really suitable for wood mouse in terms of cover. It's bare, it's open. So this is very scary. This is so any owl from above can scoop down and catch a mouse. So it takes a lot of courage to walk around here. And wood mice do have that courage. They go out and collect seeds and then hide them for later use or eat them immediately. But the bankfall, which is another mouse species here, it doesn't go out there.
Okay.
It's too scared. But in general, structure means more mice.
Literature also indicates that the presence of sufficient vegetation cover and seed-producing trees are important factors that determine the abundance of small mammals in an area. Ticks can sense these animals by different mechanisms such as transmitted heat, carbon dioxide, vibration of the vegetation, and by scent. To examine the relative importance of the previously introduced mechanisms, the following research question has been formulated. How is tick density in temperate forests related to the habitat suitability for small mammal hosts? Following this research question, it is hypothesized that tick densities are higher in forests that provide a suitable habitat for small mammals, since these animals are important hosts for ticks in their nymph stadium. Food availability and vegetation cover against predators are good indicators of habitat suitability for small mammals. Canopy cover and undergrowth can increase tick survival by maintaining a favorable, moist microclimate. A mechanism by which ticks locate areas of high host densities is by sensing host species. Furthermore, ticks will prefer locations with a structure of vegetation over locations without the structure because of the increased chances of contact with a host. From these hypotheses, it is clear that tick density can be examined on different spatial scales. The movement patterns of ticks occur on a relatively small scale of some decimeters, whereas tick densities as a result of mouse abundance occurs on a larger scale of tens of meters. In order to find the driving mechanisms that are operating on these different spatial scales, a manipulative lab experiment and a correlative field study were performed. In the manipulative lab experiment, the mechanisms that determine tick densities on small scales will be tested, with the focus on choices that ticks make on the presence or absence of host species and the presence or absence of vegetation structure. In the correlative field study, the large-scale mechanisms will be examined, in particular the relation between local density of ticks and the habitat suitability for small mammals.